Hello everyone. In this video we will show you an example of horizontal gene transfer leading to antimicrobial resistance. We'll do this through bacterial transformation of E. coli K12 using the pGLO plasmid, which contains a gene for ampicillin resistance. This will allow the bacteria who have taken up the plasmid to be selected for against those who have not. The pGLO plasmid also encodes a gene for green fluorescent protein, or GFP, which was originally isolated from the crystal jellyfish, a type of bioluminescent jellyfish that is found off the coast of North America. This is a protein that will fluoresce under UVA light. So not only will the bacteria who take up the plasmid become ampicillin resistant, they'll also fluoresce. Also important to note is that the production of GFP is regulated by an arabinose-dependent promoter region, also present in pGLO. This is important because it means that pGLO will only be expressed in the presence of arabinose. For this transformation experiment, we will need an Eppendorf tube labeled positive pGLO, which will contain transformed bacteria, and another labeled negative pGLO, which will contain a control sample not given the plasmid, a tube of LB broth, and a tube of transformation solution. This is a water and calcium chloride solution, just meant to stress the bacteria, making them competent to take up exogenous DNA. And then we'll need LB plates containing ampicillin, LB plates containing ampicillin and arabinose, and then plain LB plates containing no added ingredients. Finally, you need the pGLO plasmid, which should be kept on ice for the duration of the experiment. To begin, add 250 microliters of the transformation solution to both the positive and minus pGLO tubes. Next, use a sterile loop to aseptically transfer a colony of E. coli K12 to the positive and negative tube as well. Now use a sterile loop to withdraw a loopful of the pGLO plasmid, and this will only be added to the plus pGLO tube. Don't add anything to the negative pGLO tube in this step, as this is control. The positive and negative pGLO tubes are then incubated on ice for 10 minutes. Now, following ice treatment, the bacterial cells are heat shocked by transferring the positive and negative pGLO tubes to a water bath at 42 degrees Celsius for 50 seconds. After that, the tubes are immediately placed back on ice for an additional 2 minutes. From here, 250 microliters of LB broth is added to both the plus and minus pGLO tubes. The tubes are incubated at room temperature for about 10 minutes. Now, 100 microliters of the positive pGLO solution is plated on to LB plates containing ampicillin and then onto LB plates containing ampicillin and arabinose. Similarly, 100 microliters of the negative pGLO solution is plated onto plain LB and onto LB with ampicillin. Finally, the plates are incubated with the agar side facing up to avoid condensation at 37 degrees Celsius for 24 hours. After 24 hours of incubation, the transformed E. coli, that is the positive pGLO E. coli, will grow on the ampicillin plates and fluoresce under UV light in the presence of arabinose. In contrast, the E. coli that did not receive the plasmid, the negative pGLO E. coli, will not grow in the presence of ampicillin.